with the introduction, then of course, I am the director of the Andel Creation Research Center, which is under the direction of the Creation Research Society. Just to give you a very brief introduction, the Creation Research Society is really probably the oldest of the major creation ministries in the world, or certainly one of the oldest anyway. It was founded over 50 years ago by a group of scientists who were also Christians and believed in the biblical view of creation and felt that that was, in fact, also the scientific view as well. And so they founded this as a society for others of like mind to participate. To be a member of the society, you just have to agree with our position statement, our statement of faith. To be a voting member, all voting members in the society have advanced degrees in some field of science. And as was mentioned, I believe it was mentioned, we have recently relocated. We have a brand new Andel Creation Research Center and Operations Center for the society. We are currently located on the campus of Arizona Christian University, and this is just showing our new lobby. And we're still moving in, but it's exciting time. It's exciting to have brand new facilities, very modern facilities, and a very, very modern lab once I finally get moved in totally and up and going. And that's exciting as well. There's very few laboratories in the world that focus strictly on the biblical creation model as a scientific exploration. All right, so I do want to talk about genetics this evening, and recognizing that for most people, of course, you might as well be speaking Chinese to them, assuming you don't speak Chinese, obviously. It is certainly a field that if you don't have the training in it, it gets very complicated very fast. And we tend to just say, well, okay, and we move on. But I want to encourage you, the area of genetics is a very hot area, not only in science in general, but it's a very hot area in the area of creation science. In fact, what we're learning in genetics just in the last 15, 20 years has been wonderful for creation scientists. It has just been one after another of validating our arguments. I'm only going to be able to present just a few this evening because there's so many, but I do want to go through and just show how genetics is very much supporting the creation model and is really becoming quite an enemy to the evolution model. All right, first, I think it's necessary to define what I'm meaning by evolution. It's unfortunate in a sense that it's necessary to do that, but evolution has such sloppy terminology and such sloppy definition, which I'm going to accuse the evolutionist community of actually liking, because if you don't have to defend anything specific, there's really nothing to defend. And I think they like it where there's not really specific definitions, where it's really vague in how it's defined. But I'm going to challenge that for the most part, when they're talking about evolution, despite how they may want to define or how they want to focus, ultimately what they're talking about is what's known as universal common descent. And that is that ultimately all life shares a common ancestry. Now that common ancestry may go back to a pool of single cells way billions of years ago, but all life shares common ancestry. So that means that we and primates share a common ancestry, and we and primates and cows share a common ancestry, and we and primates and cows and trees and fish all share common ancestry. And that is the basic pinnacle position of what is generically called evolution. Anything else from that really is just some facet of it, some specific component of it. But this is where, shall we say, the rubber meets the road. This is what we're really talking about when we're talking about evolution. We're talking about the difference of conclusions between evolutionists and biblical creationists. Okay, the biblical creation model, first off, says there is no universal common ancestry. We and cows and primates and fish and trees do not share a common ancestry. Horses were created as horses, cats as cats, porpoises as porpoises, humans as humans. Now, there's been diversity of physical traits and diversity of features and such since that creation, but we don't share a common ancestry like the evolution model would portray. So there's very significant difference in the two models, and that means also very significant difference in the predictions and the expectations of the two models, what they would expect to find in the genetic data, for example. Okay, so we begin our journey, of course, we go back to Darwin. Now we got to recognize Darwin was not the first one to propose a lot of the ideas of evolutionary descent, but he certainly is the one that's given credit for kind of solidifying a lot of the ideas and also bringing in ideas of how these changes could have occurred. The problem is that Darwin knew nothing about genetics. Now, that's not his fault. Nobody at the time of Darwin's life knew anything about genetics except an Austrian monk working with pea pods, pea plants, and nobody understood his work. So Mendel's work went really unknown or certainly unused for at least 50 years, if not more. So during that time, Darwin was free to speculate and everyone else was free to speculate because they didn't know how genetic changes occurred. So we're free to speculate. We can say anything. And Darwin assumed that changes are unlimited. If you see a change of an organism going from one color to another color, that also means it can grow legs, it can grow wings, it can grow feathers. In other words, change is unlimited. Whatever change you see, it can keep going and going and going. And that's because he had no idea how change occurred, so why limit it? And if it's unlimited, then of course we can have universal common descent. We can have evolution of anything we want. So I want to actually turn to Richard Dawkins. Richard Dawkins has written a number of books. The only one he's really written that's any good for the most part is Climbing Mount Improbable. Climbing Mount Improbable, he actually, I think, outlined the situation very well. Now, his conclusions, I think, were basically erroneous, and we'll talk a little bit about some of the generic conclusions as we go through this, but I think he outlined the situation fairly well. So I want to use his illustration of climbing a high mountain to explain what I'm really talking about when I'm talking about the difference between what's predicted by the creation model and what's predicted by the evolution model. Okay, as Dawkins described it, we can look at biological complexity like a high mountain. If you're at sea level at the very bottom, that's very unsophisticated. There's virtually no biological complexity at all. And then of course, the pinnacle of the mountain would be what we'd see today, the very essence of biological complexity that we find appearing in the biological world today. And so what Dawkins said is, okay, how are we going to get from way down there to way up there by an evolutionary process? And he admits that only God would essay the mad task of leaping up the precipice in a single bound. Any designer capable of constructing the dazzling array of living things would have to be intelligent and complicated beyond all imagining. And that probably is the most correct statement that Richard Dawkins has ever made is yes, it'd have to be the most intelligent and complicated beyond all imagining. But notice, see how Dawkins admits it. If you're going to go in a single bound from the bottom all the way to the top, only God can do that. Only a creator can do that. If you're going to try to do it naturally, you can't do it that way. You got to seek some other mechanism. So that, of course, is what he's trying to seek is, okay, rejecting God, because obviously he's an atheist, but rejecting God, how would this occur naturally? How are we going to get from the bottom of this mountain to the top of the mountain by some kind of natural process? So his solution is that Darwinism solves the problem by breaking the improbability up into small, manageable parts, smearing out the luck needed, going round the back of the Mount Improbable, and crawling up the gentle slopes inch by million-year inch. In other words, he's envisioning that on the back of the mountain, it's actually a nice leisurely stroll. It might take you years and years to do it, but it's a nice leisurely stroll. You don't even hardly know you're going up the mountain. Okay, that's what he's suggesting. Now, I find it interesting, first off, that we look at, he is concluding that the way we do this is through random mutation and natural selection. And so he's concluding that if random mutation and natural selection can get you to the top of the mountain, then random mutation, natural selection actually replaces God. That's your God now. We don't need God, we got random mutation and natural selection. Waddington in 1960 stated that Darwin's major contribution, as is a major contribution to understanding origins, was, of course, the suggestion that evolution can be explained by the natural selection of random variations. The fittest individuals in a population, defined as those which leave the most offspring, will leave the most offspring. And then he goes on to say, this is previously unrecognized relation. Oh, really? So we didn't know that those that leave the most offspring leave the most offspring. So it just always never underestimate the enormous power of a tautology as a weapon of explanation, because it's a tautology. Obviously, those that leave the most offspring will have the most offspring, you know. So it doesn't explain anything. And that's obviously one of the criticisms that creationists has offered in natural selection, or at least the evolutionist use of natural selection for decades, is when you put it in those terms, you explain nothing, you offer nothing. It has no scientific validity or value to it. Okay, according to Dawkins, again in his writings, starting with climbing Mount Improbable, natural selection will sort through all the traits in a population and favor those traits that are best suited for survival. So in other words, according to Dawkins, okay, natural selection means that if you can run a little faster, swim a little farther, fly a little higher, hit a little harder, dig a little deeper, if you have some slight advantage in some action over your neighbors, over your friends, then you'll be a little more likely to survive. Therefore, you'll be a little more likely to populate, and your genes will be a little more likely to dominate in the population. And we repeat this over and over and over till supposedly at some point in time, some creature that was swimming in the ocean that slithered up on the land decided he liked the land and grew legs and became an amphibian or became a reptile, depending on your view of evolution as to if you had to go through amphibians to get to reptiles or not. So with that in mind, then let's look a little bit at what natural selection can and what natural selection cannot do. Okay, in essence, this is what evolutionists are asking natural selection to do. In some manner, to guide the process, the blind process, no less, of converting a car into a Boeing 747. When you're saying that a marine creature jumped up on land, grew legs, grew skin, you know, began to walk, then grew feathers and wings and began to fly, what you're really talking about is a transition that's actually even more so than going from a car to a 747. So evolutionists are telling us the process of natural selection is able to guide the blind alteration or changing of parts to go from a car to a Boeing 747. Now, problem number one, natural selection has no plan. It doesn't know it's trying to make a Boeing 747. All it knows is it has a car. Okay, so if I have a car and suddenly the car sprouts a wing, the actual situation is natural selection is going to favor stasis, not change. It's going to say, wait, that wing makes you a worse car. You're now going to be out of balance with your environment. Therefore, you're not favored in this environment. So the car that starts growing a wing, in fact, gets selected against by how the evolution is saying natural selection would work as a filtering process. So we really come to a point where it becomes virtually unimaginable to know what kind of changes you could make to a car step by step that natural selection would say, oh, this actually favors, you're actually now a better car, because natural selection wants to keep you a car. It doesn't have in mind, as I said, that you're needing to move or transform into an airplane. So all the intermediate steps really become less advantageous. You're less in balance with your environment and you'll be selected against. And that brings us to point number two. Natural selection cannot create anything. It only can select against. It can't even select for. It's simply a calling event. In other words, if you don't survive as well, then natural selection will say, you're out of here. Now, what's interesting is most biologists acknowledge that natural selection cannot create, and they acknowledge that it doesn't select for, it actually selects against. But then when you listen to their interpretations of their descriptions of evolution, you begin to say, okay, that sounds like it's at odds with what you just acknowledged is that natural selection cannot create. So I think evolution forces them to kind of live in a very strange contention where they're trying to acknowledge the truth that natural selection doesn't create, but they're still needing it to have some creative capability in order to achieve what they needed to achieve. And certainly Darwin didn't understand natural selection cannot create, and he very much thought that it could in fact create. So that really created a incorrect conception on his part when he was trying to explain the actions of natural selection. So we recognize the trait must already be present in the population. Natural selection can only eliminate the traits that are there. It can't put a new trait in. Let's look at the famous peppered moth study that has been around for decades, graces all of the evolution textbooks. There was a time in London, well, in the UK, but specifically in London, where you had two types of moths, a black moth and a white moth. And the white moth was by far the dominant numbered moth because a lot of the buildings and a lot of the landscape and a lot of the vegetation, trees and such were a lighter color. And the dark moth stood out. And so the thinking was that, of course, that makes them more vulnerable to predation because they're not camouflaged as well as the dark moth. Then when industrial evolution comes in and you get more soot and more smoke and it darkens the side of buildings and darkens the side of trees and such, now suddenly the dark moth has better camouflage and the white moth now is more vulnerable to predation. And in fact, this probably is a legitimate explanation in the large scale of what would happen, but it always comes back to what does this really mean from an evolutionary perspective? I had a undergraduate student in my lab one time working for me, and she was taking an evolution class. And so I from time to time would ask her a question just out of curiosity. And one day she told me that they were studying the peppered moths. And I said, oh, that's very interesting. Were you aware that, remember, both moths were there at the beginning? It wasn't that the white moth became a black moth. And she stops and she looks at me and she says, well, then what was the point? What they trying to demonstrate? Now, I want to make it clear, I doubt that the professor told him that the white moth evolved into a black moth. I'm pretty sure he probably didn't. I'll give him credit for probably not doing that, but you have to recognize what the students heard, because for them, for this to really make much sense, for it to really have much value, it has to be that the white moth evolved into the black moth. Otherwise, if they're both there, what's the point? What have you explained? And the answer is you haven't explained anything. You've explained how, in a given situation, one population or one trait is now favored, and another condition, another trait is favored, but the traits are already all there. So again, that's natural selection certainly acts in this manner, but it doesn't have any ability of doing anything beyond what's already there. Okay, if we look at the Galapagos finches, Darwin's finches, the assumption has been that it took some 2 to 3 million years of evolution for these finches to diversify into the types of beak sizes and head sizes and distribution along the islands that we see today. What's interesting is that in 1967, a study was done where they took some finches, they released them on an island that didn't have any finches. And they came back 20 years later and they found that the finches had spread to other islands, and they saw the same type of variety of beak size and beak shape and head size that they saw in the Galapagos finches. But this occurred only in 20 years, not 2 million years. And so certainly explanation is that these birds are already programmed by the creator with the ability of such diversification depending upon the type of food source, whether it's raining and the seeds are large, whether it's dry, the seeds are small. They are able to adapt to the type of food source by changing their physiology, by changing their anatomy, essentially by changing their beak shape, their beak size, their head size to accommodate. And they do this very quickly. It's not a matter of having to evolve over millions and millions of years. Right? And so we see then essentially that the shape and size of the beak is very much dependent on if it's a dry year or if it's a wet year. And it just oscillates back and forth. Nothing is evolving into anything else. This is just simply a variation. Now, evolutionists love to call variation evolution, but to me, that's just they're trying to legitimize evolution by calling it variation. But again, what they're really trying to demonstrate is universal common descent. Okay, so third, natural selection simply cannot do what evolutionists are asking it to do. And there's a lot of examples. We'll give one case in point, and that will be the honeybee. The workers, for the most part, divide into two groups. There are the proactive foragers and the reactive foragers. And what happens is that the beehive has to send out a certain number of proactive foragers to go find the flowers. Once found, they signal to the rest of the hive where they are come and get them. So you have a situation where if you send out too many proactive foragers, there's not enough of the reactive foragers to make a very large harvest once the flowers are found. But if you send out too few proactive foragers, they may spend all day and not find any flowers at all. And then the hive suffers from that. So the idea is you got to send out just enough to maximize your probability of finding the flowers, but also reserve enough that you can make a significant contribution to the hive then once the flower is found. So it really becomes a division of labor. And so a study was done several years ago actually studying the number of proactive versus reactive foragers in a beehive, and lo and behold, it's exactly mathematically the correct number. They send out just enough proactive foragers to maximize the probability of finding flowers, but have enough reactive foragers left to maximize the return on the flowers that are found. And you have to understand, if natural selection is the one that did this, it had to select through every single possible combination of number of proactive versus reactive. And that's an enormous number. And I assure you, natural selection would have been far lost in trying to do that. The hive would have been dead many times over by the time it finally stumbled on the right combination of proactive and reactive foragers. So here we see it's a situation that's really beyond the mathematical capability of natural selection. How did the bees know how many proactive and reactive foragers sent out? Because they were programmed by their creator, because the creator knew how many to send out. It's not a matter of somehow running the slot machine time after time, trying to figure out, because in so doing, the hive dies off long before you get there. All right, so we've determined that natural selection, first off, can't make anything. You can only select and eliminate. And even then, its elimination process is a bit weak. I think one analogy has been put forward. It's like the princess and the pea. You really don't have that much sensitivity to natural selection as you would need for it to serve the purpose that evolutionists want it to serve. But it comes back to, okay, if natural selection is not the one that's generating the traits, it's only selecting through the traits, then where do you generate the new traits? How do you get the wings on the car? How do you get the legs on the fish? And that's through a process of mutation. Mutations then are seen as the main mechanism for driving the stepping or the climbing up the mountain. Okay, so very quickly, we're going to define mutation as any change to the nucleotide sequence of DNA. If you remember your biology, DNA is 4 nucleotides, adenine, thymine, guanine, cytosine. They're abbreviated as, of course, TACG. When you change one of those nucleotides and that change becomes fixed, in other words, it becomes permanent, it's not repaired by the repair system, then that becomes a mutation. Now, the consequence of that mutation can be a lot. It can have very little consequence. It can have consequences that would potentially make the organism maybe more fit, less fit, neutral. And that's what a lot of this study in mutations is about. Well, what effect did the mutation have? What did it do to the organism that altered it? Did it change its color? Did it make it faster? Did it make it slower? Did it make it taller? Did it make it shorter? What did the mutation do? All right, so when we go back to universal common descent, then the idea is that through the filter of natural selection, the mutations have provided us, or beneficial mutations have provided us then the mechanism to create the changes so we can get this kind of diversity, this kind of transformation occurring. So the fish becomes some type of land creature, which becomes a reptile, which becomes a bird, which becomes a mammal, which then ultimately becomes a primate and then becomes a human. So we have mutations, specifically beneficial mutations, in other words, mutations that are supposed to be good for us, that are occurring all along the history of life, changing us ever upward in this evolutionary panorama, if you will, to give us the life or the diversity of biology we see today. So that is the power that's being invested into beneficial mutations. Now, I need to introduce a couple terms very quickly. In biology, when we talk about a phenotype, we're talking about the physical traits, the physical characteristics, your hair color, your eye color, your height, whether you have 5 fingers, whether you have 2 arms, that's your phenotype. The genotype then is the genetic system behind that phenotype. Now there's a relationship of the two, but obviously we're still learning some of the relationship. We're finding out that there's very interesting situations of certain genotypes giving certain phenotypes and not giving other phenotypes. So that's still part of where the field of genetics is at. But the genotype refers to the genetic system in the organism. The phenotype refers to the physical characteristics of the organism. And the reason that's important to understand is because I'm going to challenge that all too often in the literature, in the scientific literature, when they talk about beneficial mutations, they are almost exclusively identifying and categorizing them by their phenotype with little or no regard to their actual genotype. In other words, okay, you got this phenotype, you got this change that occurred in the organism, but what happened at the DNA level to bring about that change? And let's look at some examples to understand what I'm getting at. Okay, malaria resistance. Obviously, we've heard of sickle cell anemia as a way of resisting malaria. Of course, that's a pretty drastic way to resist malaria because you may not die of malaria, you die of sickle cell anemia instead. So I'm going to offer the evolutionist community a much better mutation, and that is the Duffy mutation. You have on your red blood cells a protein called the Duffy antigen or the Duffy protein. And the parasite that causes malaria will bind to that Duffy protein, and that's what helps it get into the red blood cell to give you malaria. But if you have a mutation that eliminates that Duffy protein, so the red blood cell does not carry the Duffy protein, the parasite does not enter in the red blood cell very well, and you are resistant to malaria. So is that a beneficial mutation? If you're in an area where there's a lot of malaria, it absolutely is beneficial, very much so. But look at what's happening at the genotype, not the phenotype, but at the genotype. At the genotype, you've had a mutation that has inactivated an already pre-existing gene. Now, any mutation that inactivates a pre-existing system does not give you an explanation where that system came from to begin with. So this may drive diversity, but it does not drive climbing up the mountain. You don't go up the mountain by eliminating stuff. That's stepping down on the mountain. That's going backwards. So if we want to go up the mountain, we got to have mutations that are going to go upward. We're going to have mutations that are going to explain how we got this to begin with. Okay, let's do another. DDT resistance in insects. I see this in textbooks all the time. Evolution of insects. They became DDT resistant. Well, there seems to be a number of very specific ways that insects develop DDT resistance, but in general, a mutation is introduced into a regulatory component of the CYP6G1 gene. That gene is responsible for making a protein called P450. And the mutation creates an excess amount of P450. It doesn't eliminate the making of the P450, it makes an excess amount of P450, which is what contributes to the insect's DDT resistance. All right, let's look at this again. At the phenotype level, beneficial, the insect resistant DDT, it still lives, it's evolved. Okay, but at the genotype level, what has happened? A mutation has wiped out the regulatory control of this gene so that it is now overexpressing, is making too much of the P450. So this mutation cannot explain not only where the P450 gene came from, but it can't explain where the regulation of this P450 gene came from because the mutation knocks that out. So again, we're knocking out a pre-existing system. It's giving us a beneficial phenotype, but the genotype is we're eliminating pre-existing systems. Another example, HIV infection. If you are exposed to the HIV virus, it binds to a protein called the CCR5 protein, which is on one of your immune T cells, one of the cells part of your immune system. Okay, and that virus then, once bound to that CCR5 protein, will be able to infect that cell, and potentially you would potentially be able to get AIDS from that. All right, now there is a small population of people that have a mutation in the gene that makes that CCR5 protein, and they no longer have it on their T cell. And therefore, the HIV does not bind or infect their T cell, and so they are in effect resistant to HIV. Now, beneficial phenotype, absolutely. If you're exposed to HIV, it's nice to be resistant. But let's go back to the genotype again. What's happening at the genetic level? The genetic level, we have knocked out a pre-existing protein. So this mutation doesn't explain where the protein came from to begin with. It only explains how we knocked it out. So again, that's not stepping up the mountain, that's going backwards. That's stepping back down off the mountain. And human lactose tolerance. Most children pre-puberty make a enzyme called lactase, and they're able to digest the lactose of milk. But as they pass through puberty and become an adult, there is a regulatory system that kicks in that shuts the lactase gene down, and they no longer make the lactase enzyme. Therefore, they cannot digest the lactose in milk, and they would often be what we'd say have a milk allergy. Now, this is actually most people, or the majority of people in the world, are lactose intolerant. They do not make that lactase gene after post-puberty. But there are some of us, especially those of us that have more of a Northern European ancestry, or at least some of our ancestors were Northern European. Some of us have a mutation in the MCM6 gene, which is involved in regulating the lactase gene. And that mutation keeps then it from shutting down the lactase gene, and you continue to make the lactase enzyme through your adult life. Now, is this beneficial? Well, I'm glad I have this mutation because I like milk and I like drinking milk, and I'm glad as an adult I can drink milk. So yes, it's beneficial if you want to drink milk, but once again, let's look at it. Genotype-wise, it's a mutation that knocked out a regulatory system. So this mutation does not explain how you climb Mount Improbable. It only explains how you step backward off Mount Improbable. In other words, losing systems that are already there to begin with. Antibiotic resistance. It's amazing to me that as creationists, we have to keep talking about this, but the evolutionists, they persist. They keep it in their textbooks. They keep referring to it all the time as examples of evolution in action. Well, again, it's an evolution in action only if you're defining evolution simply as change. But if you're going to the deeper component of saying, but evolution, ultimately you're looking at is universal common descent, how does antibiotic resistance explain that? And the answer is, of course, it doesn't, because it's not providing that kind of genetic mechanism. Antibiotic resistance says that you have a population of bacteria and they're exposed to an antibiotic. Those members of that population that have a resistance in some manner by some mechanism will survive this antibiotic. So certainly for the bacterial population, or the cells that survive anyway, it's very beneficial mutation. They've lived through the antibiotic to live to fight another day, if you will. But let's look at what is a common cause or a common reason for antibiotic resistance. Okay? Loss of enzyme activity, halting of cell division, loss of regulatory protein, reduced formation of a poron or regulatory protein, loss of a poron, loss of reduced, loss, reduced, loss, reduced, loss. Do we see a trend here? See, the bacteria are becoming resistant to the antibiotic by essentially eliminating some system that either allows the antibiotic to get into the cell or allows the antibiotic to interfere with cellular systems in the cell. So by eliminating that, then the antibiotic won't have an effect on the bacterial cell. And as such, they can survive the presence of the antibiotic and live, as I say, to fight another day. It's a little bit analogous to your foot is caught in a bear trap and here comes the bear. What are you going to do? Well, see, you may have to decide you got to chop off your foot, although that still may not get you very far, but you decide you have to chop off your foot. The advantage bacteria have is they can chop off their foot but grow it back. You can't grow your foot back, but they can. Once they lose the system, they survive the antibiotic onslaught, but now the antibiotics are removed from their environment. They then go back to actively making that protein, actively making that transport system, actively making that poron. So they have that advantage that you don't have, and that's why they can respond very quickly to very harsh environmental conditions. But once again, let's put it in perspective. What is happening that's giving them the ability, mutation-wise, to be resistant to the antibiotic? It is a mutation that is eliminating pre-existing systems. It is in no way giving the antibiotic some new characteristic, some new gene, some new genetic event that it did not previously have. We're eliminating pre-existing systems. So we're coming back down off the mountain. We're not climbing up the mountain. Okay? The long-term evolution experiment, I would assume many of you have heard of this. This is an experiment that was done at Michigan State University by a professor named Linsky and his graduate students and such. And it's a very good experiment. I love the experiment. I wish I would have thought of it. It's a great experiment for microbiologists, but I would argue that that's all it's really for is microbiologists. But he took a strain of E. coli and he cultured it day after day, week after week, year after year for over 65,000 generations. And one of the key things you have to recognize in this experiment is the media and the temperature were always the same. In other words, the E. coli never saw anything different for years and years and years. It was the same thing day after day after day. What they found was, yes, they began to get spontaneous mutants that have formed in the E. coli. Some of them were, for example, the E. coli started knocking out DNA it didn't use to shorten the chromosome so it could replicate faster. Okay, but that doesn't explain where the portions of the chromosome that got knocked out came from. It just means that the E. coli found that, hey, if I have a mutation that inadvertently deleted part of my chromosome, that's actually a benefit because I can now replicate faster. Other mutations eliminated various metabolic pathways and such that the E. coli wasn't using. So by and large, all the mutations they found were mutations that Linsky identified as what he called antagonistic pleiotropy. In other words, it's a trade-off, a sacrifice of a particular existing system that's not essential in a specific environment if it helps increase the adaptation to that specific environment. I don't need this DNA in this environment, so I knock it out, and lo and behold, it actually serves an advantage to me to get rid of it. And so what they saw, for the most part, in all of their mutants was, with just a couple of exceptions, is a trade-off where it would eliminate systems that it didn't need, it didn't use, which wound up allowing it to grow faster. It wound up allowing it to adapt better to the particular environment. So again, if we look at genotype versus phenotype, yeah, we have this phenotype where it's growing faster. This mutant now grows faster than its original parent does, but let's look at the genotype. It grows faster because it got rid of a lot of stuff. It threw things overboard that it didn't need in that specific environment. Now, the moment you took any of these mutants out of that specific environment and put them into a different environment, obviously they didn't do very well. But of course, that wasn't the point of the experiment, but the way that they're interpreting the experiment, we always have to be reminded of that this is a very environment-specific mutation that occurred, a very environment-specific condition, and you change the environment and suddenly it's no good to you anymore. All right, so to kind of back up from that then, let's put it into an analogy, beneficial mutations. I call it the analogy of the house and the happy wife. All right, let's say in your house, you have the dining room and across an interior wall, then you have your rec room. Because your wife is, the socialite of the town and she loves throwing big parties and having a lot of guests over and cooking big meals, she announces to you one day she wants a bigger dining room. And you get thinking about expanding the house and look at your checkbook and decide that's probably not going to work, probably can't pay that price. So it becomes a lot easier to knock out an interior wall, getting rid of your rec room, but now you have a much, much bigger dining room and the wife is happy. And of course, if the wife is happy, then everybody's happy. And so it's a beneficial mutation. But how was it done? It was done by knocking out an interior wall. What evolutionists have done to us in all their literature talking about this mutation and that mutation, and you'll notice the examples I gave you, these are the classics. There's all kinds of them all over in the literature, but these are the ones you're going to find in the textbooks. These are the ones that are going to be being taught all the time. These are the classics. Okay, and what they're trying to give you the idea of is these mutations somehow built the wall. The reality of it is, no, they destroyed the wall. They knocked the wall down. So to me, when they give you these examples, it's analogous to you telling a carpenter the way you built this house was by knocking out this wall. And don't be surprised if the carpenter kind of looked at you funny and said, you really are pretty stupid, aren't you? Okay, so bear in mind, see, when evolutionists are telling you that knocking out the wall somehow made the bird, somehow made humans, recognize they're completely missing the point of what happened at the genotype level. So what is it we observe actually occurring when we look at the types of changes that occur in biology? See, which direction are we going? Are we climbing up the mountain, or are we actually descending? Well, these are knocking out. We're going down from a pre-existing complexity, a pre-existing system. See, as a creationist, we would argue that God placed us at the very top of the mountain, and everything that's happened since then has begun to move us down off the mountain, physically speaking. Okay, but it doesn't stop there. There's many more problems that the evolutionists have to face as far as genetics is concerned. Okay, there was a study in 2018 where they looked at 100,000 different species and they analyzed their mitochondrial DNA. Now, very quickly, mitochondrial DNA is DNA in the mitochondria of the cell. That's a small organelle in the cell that's separate from the chromosomal DNA. Okay, they looked at the mitochondrial DNA of 100,000 different species, and they found that 90% of them originated at approximately the same time, which is not consistent with universal common descent, which would say you should be getting species occurring over a long scale period of time, and they should be appearing and disappearing and appearing, disappearing, and yet we'd say all contemporary species for the most part appeared all at once. See, that's more biblical. That's certainly not universal common descent. Also, Fisher's fundamental theorem of natural selection. I remember studying this as an undergraduate when I took an evolution class, and I have to admit I didn't have the mathematical capability to really digest it, but it always seemed to me there was something wrong with his conclusions. I could never quite put my finger on it, but it seemed like something wasn't right with how he went about it. Well, as I studied it more, began to recognize that he defined change of mean fitness will equal genetic variance of a species. In other words, if you change the fitness, then there's going to be genetic variance of the species. But in this, what he did was he assumed that all mutations will continually increase the fitness of the population. In other words, it doesn't make any difference what mutations occur. So he didn't worry about that. It's just as long as they continue, that's all he's worried about is mutations need to continue. He defined then they will increase the fitness of population because they're going to increase the genetic variance, which means they're going to increase the mean fitness. And when I saw that as an undergraduate, it's like, there's something not right with that, but I couldn't lay my finger on that. Well, since then, of course, work has come out that we now recognize that that's in fact not true. Continual occurrence of mutations actually will cause a decline in fitness. It'll cause a decline in fitness in any population. And this, in fact, contradicts Fisher's assumptions. And this was published in the Journal of Mathematical Biology in 2017. So while Fisher was offered and is still offered today as a mathematical verification of Darwin's arguments, it's actually not true. He made the wrong interpretation. And in fact, what we know about mutations is the exact opposite of what Fisher was assuming. There's another problem. It's called the waiting time problem. Simply put, there's not enough time for all the genetic changes that separate us from primates and our common ancestors. In other words, between we and chimpanzees, there are so many genetic differences that there's not enough time in these 6, 7, 8 million year period, supposedly from our common ancestor today, for all these genetic differences to have accumulated, to have occurred and accumulated and become fixed. And this is a well-recognized problem. Evolutionists have been discussing it for years, trying to figure out their way around it, if you will. They can't figure their way through it. They have to figure their way around it. But it's a very real problem. You cannot fix mutations fast enough in a population to be able to explain all the genetic differences you're going to see in different populations that supposedly share common ancestors, 3 million, 5 million, 10 million years ago. You just don't have the time to do it. So we would have to recognize we're in the 21st century. See, if life was simple, then evolution's mechanisms of mutation, natural selection would work, or they likely work in a way. They may well work. But see, life is anything but simple. In fact, we now understand life is extraordinarily sophisticated. Hence, Dawkins' view of life's complexity is that, scaling the tall mountain. Okay, so we now recognize that because life is anything but simple, we're asking so much out of mutations and natural selection that they simply don't have the power to deliver. They do not have the power to climb that mountain that evolutionists require them to climb. They don't have the power to go up the backside as evolutionists are trying to claim that it does. There is no gentle slope that you keep moving up. It doesn't exist. The idea is you're going down. Mutations take you down, they don't take you up. This is well recognized, not just by we creationists. There's a number of evolutionists recognize this also. And that's led them to search for what they call the third way of evolution. They recognize that contemporary views of evolution as far as mutation and natural selection does not work. So they're looking for a third way. There's got to be another way. There's got to be a way that evolution will work, that'll climb the mountain, that doesn't involve God. Now, they're still looking. They haven't found it yet. But this is an illustration of what I'm talking about. They would agree with most of what I said. They would say that, yeah, you can't climb the mountain through mutation, natural selection. There's got to be another way. And we're going to look for that other way. But in the meantime, I think that by far the most consistent scientific explanation is we were put on the top of the mountain by the creator himself, not trying to scale the mountain by some process that doesn't work. Remember, as Dawkins even admitted, to get to the top of the mountain, you either have to scale it by a process that I'm saying doesn't work, or God places you there. And to me, scientifically, it makes much more sense that God placed you there rather than relying on a mechanism that scientifically doesn't work. So with that, I will close and we will open it up for questions.
Thank you very much, Dr. Kevin Anderson. Some great highlights, some new things, I think that we've been seeing, learning about. So it's always great to be able to hear new things. And we'll have people, if you want, either there's a Q&A feature in Zoom if you're joining us via Zoom. If not, if you're on Facebook Live, feel free to enter that through our comments on the event, and we'll try to collate those. So I appreciate the people, everybody who's tuned in. Hope you could see that when we look at science from kind of the present day, looking at what we can actually observe, not what usually turns out to be kind of like fairy tale storytelling, more or less. And yeah, we see that it does line up with what would be infinite designers and created us with all the information from the start. And since the fall, since man sinned and rebelled, we've been going downhill. I think very eye-opening for me, especially that difference in physical characteristic traits, which is what beneficial mutations may lead to, changes that we can observe there. But when we dig deeper at the genetic coding, we find that essentially every single case we look at the details, it's actually backwards, would be really maybe described as devolution. So we'll yeah, start off if people already have. Just a comment here. You've discussed deletion mutations in detail. I don't think that was a question, but maybe just a comment. If not, feel free to put that in, frame it as.
And of course, not all the mutations I explained were deletion mutations specifically.
Yes.
They're just simply mutations that eliminated a pre-existing system. Specifically, deletion mutation is where you actually remove some of the segment of the DNA. But you can delete a gene simply by causing a change in its promoter site or causing a change somewhere in the open reading frame of the gene that no longer is able to make a functioning product. Mm-hmm. So
yeah, so I think this person didn't have the finish her. So first question.
I mean, some of the mutations, in fact, would be addition mutations,
right?
Where you actually insert a transposon and it eliminates then the regulatory site. So it would actually specifically be a insertion or addition mutation, but as such, it's still, the effect of it is that it eliminates the functioning of that system, of that site. So there's a difference between the actual mechanism of the mutation, whether it's a missense, whether it's a nonsense, whether it's a insertion, whether it's a deletion, and the consequence of it, which is eliminating a regulatory system or eliminating a functional product of the gene. So.
Yeah, thank you, Dr. Kevin Anderson. The person, was kind of asking if there are any, have scientists not found any examples of addition mutations? So it's good to explain that they still turn out to be essentially negative.
Now, Gerald here asked a good question. I'm going to take a different perspective than a lot of my creationist colleagues.
Yeah, maybe you can say,
mutations are a loss of information. I'm not going to say they necessarily are, because the problem we have, we don't know how to define biological mutation, let alone genetic mutation. And therefore, I am unwilling to say that it's a loss or a gain, because we don't know how to define it, let alone measure it. And if I cannot measure it, then how do I know if it's a gain or a loss? So I think that it's incorrect for creationists to talk in terms of gaining or losing information, because we can't define it that well. We don't have a measurement of genetic information. That's why when I talk about it, I talk specifically at what's happening at the DNA level. Did we eliminate a pre-existing system? Did we create a loss of a functioning protein or a loss of a functioning, enzyme? That's why I do it that way, as opposed to ever talk about, well, we lost information. I don't know if we lost information or not. If you've ever read student essays, you recognize sometimes knocking out words actually increases information. So I'm not going to say that mutations lose information. I'm simply going to say mutations do not give us the genetic mechanism that allows us to climb Mount Improbable.
Yeah, I guess you're saying there's got to be increases in function, increases in structure, these sorts of things.
And even more precisely than that, there has to be a genetic event that demonstrates how you made a transport protein, that demonstrates how you constructed the particular structuring of the DNA so you can have like chromosome kissing, those types of things. All these are events we observe in the cell all the time. And I'm asking the evolutionist community to explain to me through their process how these came about. And that's what I'm still waiting on, is they haven't been able to explain yet. To me, it's more than a matter of loss or gain of information. It's a matter of what's your mechanism of explaining what we're seeing going on in the cell. Mechanism of explaining the origin of what we see going on in the cell. And that's where I say they haven't yet been able to deliver.
Mm-hmm. Yeah, there's lots of questions coming in. Thanks, everybody. Okay. Keep them coming in. We'll take
all right. Epigenetics. All right. Yes. Epigenetics is one of the greatest things to happen to creationists. Oh my goodness, epigenetics. I can't even hardly get evolutionists to talk to me about epigenetics. They'll spin some kind of story to try to explain it, but oh my goodness. If it was tough enough for them to be able to explain how changing a thymine to an adenine suddenly made some difference that allowed universal common descent, you've got to take it up a notch, because epigenetics is the regulatory aspect above that sequence, by methylating, by not methylating, by acetylating, by not acetylating, by putting a certain twist in the DNA, by taking that twist out of the DNA. So you're looking at a much, much more sophisticated language than just the A's and the T's and the C's and the G's. It's tough enough to use the A's and the T's and the C's and the G's to try to explain mechanisms to account for universal common descent. But if you're now also trying to include into that the much more sophisticated language of epigenetics, honestly, what they're offering to try to explain it, I think it just shows that they're not able to really come up with anything. It's a lot of speculation. Well, maybe this and maybe that, and could have been this and could have been that. Shapiro's been one of my favorite writers over the last several years. He's one of them, in fact, as a member of the third way of evolution. He recognizes that you cannot account for what we see going on in the cell through the natural processes that we observe through mutation natural selection. And what amuses me is because he's not going to accept that the creator made it, he's still got to come up with a mechanism to account for it. And all these mechanisms still comes back to mutation natural selection. So mutation natural selection made a system that now cannot be explained by mutation natural selection. So there's a certain inconsistency. And yes, he didn't use that terminology, but that's still what it comes down to. He needs some natural system, which ultimately involves changing the DNA, and then that DNA then create some kind of informational program that allows something else to occur. So it's all still back to mutation natural selection. Mm-hmm. So, and again, that doesn't address epigenetics. Epigenetics is the whole informational layer beyond that. There's a lot of epigenetics that we don't even understand yet. And we're finding in humans that you can inherit, if your parents smoked, that can be a problem to your children, especially if it goes through the mother. That can be a problem. You know, if they were obese, if they had a poor diet, that can have resonating effects down generations from there because of the epigenetic tagging that goes on. And so these are all things we're still getting a handle on, but oh my goodness, what it really means with epigenetics is the mountain has just gotten higher, is what it really means. The mountain has just gotten higher. And the ability of climbing that mountain for the evolutionists is even more daunting than it was before.
Great, thanks. I think there's a good one about the early population, whereas from a biblical perspective, there'd be only 2 people. So can you explain why many geneticists have a hard time postulating the existence of an original pair? They always say there's going to be minimum population size in the thousands. And what is the mitochondrial evidence that points to that original pair? So what's the story with the genetic variants that old earthers have a hard time explaining from a young earth? Also, a separate question there about what that 2018 paper was you cited.
Okay, well, I'd have to go back to my notes, but all right, so let's go through this a little bit. Okay, why Minigenes have a hard time postulating the existence of an original pair? Well, to be honest, one of the big reasons is because they're assuming incorrectly. They're assuming the original pair would have been homogeneic. But there's no reason the original pair had to be homogeneic. In fact, they obviously weren't totally homogeneic because Eve was female, Adam was male. So there's some difference already, but there's no reason to say Eve was a clone of Adam. There's no reason to say that Adam himself was homogeneic. So if we start out where the original pair was heterozygous, where they didn't carry the same alleles on both of their chromosomes, it's not that hard to necessarily then suddenly see pretty rapid diversity occurring. A lot of times, the reason that geneticists are very resistant, if you will, of starting with an original pair is because, in all honesty, it sounds too biblical. You know, if we go back to mitochondrial Eve, where in the 1998 paper in Science, they were postulating or pondering all of the ramifications of the models used to study mitochondrial Eve, and was recognized that if we just strictly take the mutation rate of mitochondria, Eve becomes 6,000 years old. And of course, oh, that's anathema. I mean, that's unheard of. So it's too biblical. So anything that has that kind of tone to it, they immediately react to negatively, in that, well, obviously there's something wrong here. This is not allowable. And so the idea of original pair, no, that's just, we're not going to allow that. Although then to explain what we see today, the evolutionist community has to constantly have humans going through various bottlenecks all the time. So they still reduce it down to very small pairs. They just don't go down to 2, but there's no reason that you can't start with 2 to accomplish the same thing.
Yeah, I heard it. Yeah, there's I want to kind of let other people have a chance to ask some questions, because we got a lot of good questions.
Yeah, well, I want to come back up here to this question by Shar. Why can't removing some of the function of a gene make a move up the mountain? And again, this is a good question, in that this is the type of thinking that unfortunately we need to get people to understand what's the problem in that. Because yes, when you are moving along a biological lineage, yes, the fish had to get rid of their gills, they had to get rid of their scales, they had to get rid of their fins. And that's all true. So part of doing that would be to get rid of, but they had to replace it. And if all you're pointing to is how the fish got rid of the gills, got rid of the scales, got rid of the fins, and you're not figuring out how it grew the legs, it grew the skin, it grew the fur, it grew the wings, then all you have is a naked fish. You know, you don't have anything. And so, yes, they do have to get rid of, but that's not the kind of change that drives you up the mountain. That's the kind of change that keeps you in the same or even going down the mountain. If you're not replacing, getting rid of the gills with something else, then you haven't gone up the mountain. All you've done, in fact, is gone down the mountain. So we have to recognize, yes, changing a car to an airplane would involve deleting some of the car function, and that's very true. But if you're not replacing with airplane function, you just have a frame of a car. You don't have an airplane or a car when you're done. And that's my contention is you're not replacing, you're not showing me how you're building the airplane, you're just showing me how you're stripping the car. And stripping the car doesn't get you an airplane, doesn't even get you a car pretty soon.
Right on. I want to skip down to someone is asking about what's the status of the argument against junk DNA.
Junk DNA, again, is a real boom for creationists in that as we study the genome, we recognize that it contains less and less of what would have been identified as non-productive or junk. Now, granted, you get into some definitions of how do you define junk and how do you define function and such, and not wanting to spend a lot of time getting into that, but the point of it is junk DNA was originally a concept proposed several decades ago when they realized that most of our genome does not code for protein. And because they didn't know anything about epigenetics, they didn't know anything about any regulatory systems, anything about, as far as enhancers upstream and such and regulators downstream. They didn't understand any of that. They just assumed promoter gene. If it doesn't make a protein, then what purpose is it? They didn't know anything about regulating non-coding RNA, didn't know anything about any of that. And so they thought, well, all we have left in our genome is about 2% that code for proteins. Therefore, the rest of it must be useless. And so the scenario is that it's useless because it's residual. It's your vestigial DNA, just like vestigial organs. That DNA used to be your gills and used to be your scales and used to be, your tail and such when you were a fish and when you were a reptile and such. And you don't chop the DNA out, but because you're not using it anymore, it randomizes because all DNA is subject to mutation. So over time it randomizes and it's simply just still there, but it's not doing anything. So that's the general concept of junk DNA. As we're now digging into the chromosomes, digging into the genome, and we're finding that there's a lot more to genetic information. And there I'll use the word information. I'm not talking about increasing or decrease. I'm just talking about there's a lot more to genetic information than just simply the A's and the T's and the C's and G's. And there's a lot more to genetic information than just simply protein coding genes. We're finding out more and more, like with the non-coding RNAs and like with the control systems, the enhancers upstream and such. So much of it involved in controlling those protein coding genes that wasn't even considered that many years ago. And so that's not junk. Just because it doesn't make a protein doesn't make it junk. And no, has function been identified for vast majority of the genome yet? No, of course not. Just like we haven't discovered all the stars yet. You're only looking at just certain ones of them because there's so many. There's so much in the genome. But each day there are dozens and dozens of papers defining new function for areas that originally were thought not to be a function. So to me, to immediately call an area junk because you don't know what it does is erroneous, because in a sense you're identifying its function. This function doesn't have a function. Well, how do you know it doesn't have a function? We haven't studied it yet. We don't know. So it should be identified as function not yet determined, rather than the fallback being, well, if we don't have a function assigned, then it doesn't have a function. Because that in itself is an erroneous assignment of function that's not correct. So most of our genome, or a vast amount of our genome, does not yet have an assigned function because we have not yet studied it. But more and more of our genome has assigned function. As I say, there's dozens of papers every day assigning new function to various aspects of various parts of our genome. So the junk DNA argument is really falling by the wayside. And it's getting to the point where evolutionists are going to have a more and more difficult time explaining without junk DNA all the arguments that they've been trying to make as far as the absorbing mutations and the genetic load that our chromosomes can handle and such. So
yeah, it sounds like evolutionary thinking has delayed us, set us back for like half a century. We could have been studying, looking at all these functions, looking for cancer treatments in areas of the genome that, yeah, they didn't know.
Right. That's an area. In fact, I would accuse the evolutionist community of slowing medical science down by decades, because until probably really, I mean, they started studying it soon after the Human Genome Project, but there wasn't an intense study of junk DNA until probably the last 12 or 13 years, maybe with ENCODE's first work coming out. And they're finding these are areas of medical importance that have gone unstudied for decades. We should have been studying them in the 80s and the 90s, and we weren't because they were junk. So we're really behind in that regard, and because of the assumption that they were junk.
Probably also related to how people make the similarity to chimps being our genome and chimps 98%, but then they don't look at the junk DNA, but to look at that evaluation, I guess, too, right? So let's see if maybe focus on the topic at hand. Can a radioactive environment, I guess, create beneficial mutations that add information? I guess maybe can you explain what does radioactivity do to our genome?
Well, radioactivity in the form of like uranium with alpha and beta particles, also X-ray could be considered a type of radiation, gamma particles, cosmic rays, a form of radiation. Ultraviolet light is a non-ionizing form of radiation. All of these have the potential to interact with the DNA and as a consequence, create a mutation. In other words, change that DNA sequence. So like anything else, then we're back to, we change the DNA sequence, we create a mutation. What's the effect of that? And then that really just harbors back to what I've been talking about in the whole presentation. The mutations don't do what they're argued in the evolutionary textbooks that they do. They don't provide the mechanism to explain how you got this genetic system. They don't provide a mechanism to explain how you got that transport system, how you got that surface, binding protein, how you got that regulatory protein. So whether the mutation is caused by radiation or caused spontaneously, the mutation itself still doesn't explain the question that I'm throwing out.
Right. Maybe we could go back a bit to I don't think this is related to the junk DNA, but can you discuss your thoughts on how many evolutionists see what they believe to be broken genes or pseudogenes, claiming to fit a pattern of nested hierarchies? I guess they say they find them in separate.
Well, yeah, the nested hierarchy. Okay, if we go to the pseudogene, first off, assumption number one is the pseudogene represents a gene that is now broken. Response number one is that many pseudogenes have vital functions. So that seems to be strange for a broken gene. In fact, I would say that the broken gene concept is an evolutionary imposement of interpretation. It's just a gene that has that structure to do what it needs to do. That its structure is similar to a different gene doesn't mean it's been that gene at one time and is now broken. It has a different function, but to carry out that function, it has some similarities to the other genes. So first off, we need to get rid of the idea that pseudogenes represent broken genes, because they don't all do. Many of them have vital functions. Second off, we don't know the functions of all pseudogenes. Again, they're in the category of not yet studied. We don't know. Maybe they have a function, maybe they don't. Okay. Third, creationists don't say that the genome is pristinely maintained since creation. Mutations have entered in. In fact, in the human population, there's an estimated 100 new mutations every generation. So mutations are entering in, they are creating damage. So we have probably lost some genes, or certainly would appear to have lost some genes. So some pseudogenes may in fact be broken genes. But the fact that we and primates may share we and chimpanzee may share a particular broken gene doesn't mean we inherit it as a broken gene from our common ancestor. It may mean that is just the nature of that gene in its function. It appears to be the broken gene because of our interpretation, but it's not. Or it could also be that it happens to be in a hotspot. There are hotspots in our chromosomes that mutate a lot. And it may be that in both us and the chimpanzee, it's in a hotspot and they both mutated. But we also have to recognize that not all pseudogenes fit a hierarchy. There are pseudogenes that are in some of the lower primates that are not in chimpanzee or not in human. See, so they don't all fit that hierarchy. In other words, we may share a pseudogene with an orangutan. Now, I'd have to, I think it's a gibbon maybe. There's some papers I'm thinking of, but I don't have it in front of me. But we share some pseudogenes with some of the lower apes that the chimpanzee doesn't have. And it might be the other way around too. It might be the chimpanzees share some with them that we don't have. I don't remember. But anyway, so it's not a nice smooth hierarchy. There are definitely some breaks in the pseudogene lineage, if you will.
Yeah, and I think there's also that case argument of the two chimp DNA chromosomes that come together, fused, and that it's equivalent to one of the human
chromosome 2. Chromosome 2,
yeah.
Because chimpanzees have more chromosomes than humans do.
Mm-hmm. And then, yeah, they say, but then if you look more closely, what would be the details of that it couldn't have been joined, say, as they do say they were joined?
Correct. If you take a much, much deeper look at the actual sequences along what's considered to be the fusion point, where supposedly somewhere in human ancestry, we had two chromosomes fused together that did not occur in the chimpanzee ancestry, so they have more chromosomes than we have. If you look at the actual sequences along this supposed fusion point, you don't find the characteristics of a fusion. It doesn't really serve that. And so assumptions have been made that it's a fusion simply to be able to present it in terms of common descent. But as even Kenneth Miller admitted, if a fusion has not occurred, and I would argue the genetics shows that there's no evidence to say it has occurred, if a fusion has not occurred, then common descent can't happen. And I'd say, yeah, I agree. You've lost because there's no fusion, and therefore you can't explain how the chimpanzee has more chromosomes than we have.
There's a question, is there a known mechanism by which an organism's DNA mutates to modify its own genetics, for example, in the case of the bacteria?
Oh, bacteria do it all the time. Mm-hmm. It's fascinating. Now, you have to recognize, and again, this is the limitation. This is one of the limitations of Linsky's work. Bacteria are asexual reproducing organisms that can grow very, very fast. Humans, for example, are sexually reproducing and we don't grow very fast. Bacteria can pay what's called the cost of selection. In other words, if you wipe out all but 2 of the bacteria, they'll still be back in big time the next day. They'll rebound and they'll be back. So if I throw an antibiotic into a big population of bacteria and only a few of them are resistant, the next day they're all back, the population's recovered. You don't do that to humans. If you wipe out most of humanity, it takes a long time for it to recover. Okay, so you can do a lot of things with bacteria that you can't do in higher organisms. Bacteria will intentionally create mutations. E. coli, for example, has an insertion element. It intentionally inserts into its chromosome for the purpose of generating mutations in its population. Random, maybe, we don't know. That's one of the questions, is how random is it? It's kind of interesting. But the point of it is that if it creates a bad mutation and that part of the population dies, so what? We can pay that price because we grow so fast. We got so many. In one flask, we can have, 100 billion bacteria in one flask. And they're growing, their generation time is measured in hours or less. And so they can recover very quickly if a mutation is introduced in the population that does not function very well. Can't do that with higher organisms. Mm-hmm. So yes, bacteria intentionally mess up their DNA for the purposes, I would interpret it, of trying to generate this diversity within the population. So they're not all clones of each other. But we also see, like in salmonella, there's a particular environmental situation that causes salmonella to totally flip a gene to turn it on. And then when it's turned off, it flips it back. And we look at that and say, what in the world is going on? See, so we're finding those kind of things in bacteria all the time where they will manipulate their own chromosome in response to environmental factors, in response to environmental events going on that isn't just a matter of turning on and off gene by the standard repressor protein or enhancer protein, but actually altering the DNA.
Mm-hmm. Yeah,
and again, because they can pay the price to do that.
Right. And I think I was thinking about along the same lines as this one person asked, what about the novel coronavirus, as I guess SARS-CoV-2? We hear of multiple mutations, which reportedly are more infectious and perhaps even more dangerous than the virus first discovered in late 2019. Is this scientifically possible that a virus replicates itself and becomes more efficient in its transmission?
First off, you have to recognize how viruses are working. And it's really a misnomer to try to, and this is where I and virologists go around and around, because I do not consider viruses living. Because if a virus is living, then so is a prion and a viroid, and they don't acknowledge prions and viroids as living, so I don't know how they acknowledge a virus as living. But a virus is a replicative entity. But the mutations that are occurring that have the medical community so concerned are mutations in the spike proteins on the surface of the virus, which is what your immune system is using to identify the virus and thereby respond to it. And it's like putting on a mask. If the virus changes that spike protein, then what happens is that the immune system doesn't see it or doesn't see it as well, and therefore its response is slower and the virus thrives better. But it really, in many ways, is just simply back to Darwin's finches. These, particularly RNA viruses, they have a very high error prone. They have a very high mutation rate. And because of that, they will make a lot of defunct viruses. But again, they can pay the price of that. But they will make also some viruses that they've altered that exterior protein on. It doesn't have anything to do with their metabolism. They don't have metabolism. It's just simply an inert protein on the surface that they use to bind with, that they use to bind to your cell with, or not. Maybe they don't even bind to the cell with them. Maybe it doesn't serve any function at all. It just depends on what the protein is. Depends on what kind of spike protein it is. But either way, what happens is the viruses go through a whole lot of different surface proteins. And it doesn't affect the virus at all because it's not serving any particular function of the virus other than just being on the surface and allowing it to bind to your cell. And if it happens to help it evade, then that's all the better for it. And so, like I say, and maybe not so much back to Darwin's finches as much as back to the black moths and white moths. These varieties exist simply because of the air proneness of the virus. It's not a matter of, quote, evolving. It's just a matter of the virus protein is mutating. But those mutations that are occurring in that envelope protein do not in any way explain the mechanisms of how the virus is making the protein or how that protein even originated. You're just simply altering that protein. If we altered proteins at the level of viruses alter proteins, we'd be dead. But see, the virus can do it because, again, it can pay that price. And the altering of this protein often is not that consequential to the virus, except, wow, hey, guess what? I can evade the immune system better. So yes, if you want to look at it as that there is an adaptive response of the virus and natural selection, quote, quote, is kicking in, absolutely. Creationists don't deny that. But if you want to look into this as an example of how organisms evolve, I'd say nonsense. It's not an example at all. It doesn't give you any mechanism that's going to explain how you climb the mountain.
Yeah, and I think it's very important how we describe, like, I like how you start out the presentation defining the term of what we mean by evolution. I think a question here related to how to best explain kind of what you're teaching more at the maybe lay level. So this person, I think, puts in a question, rather than just saying that mutations must create new genetic information, does it make sense to say that the mutations must cause genetic change that creates new previously unconceived body parts with new previously unconceived functions? So that's from Barb.
Yeah. Again, terminology becomes very important because evolutionists love to jump on various terms, but I think you're heading in the right direction. They have to provide genetic mechanisms that explain the origin of what we see, because that's the whole, they're claiming that their model explains everything. It explains all life. It explains the existence of humans. It explains the existence of birds. It explains the existence of fish. Okay. How did that happen? How did you get humans? How did you get birds? How did you get fish? Give me the mechanism. Say, well, then I get mutation natural selection. I say, I'm sorry, I don't think that gives you that. I don't think mutation natural selection is going to give you a human or a fish or a bird. So yes, it is, if you want to be very precise about it, it is a matter of the mutation has to be able to give you something that is causing the cell to have, and I don't want to say a new gene, because again, the terminology gets so sloppy because genes themselves we can't even define anymore. But when I look at the mutational event going on, is this mutational event now able to explain where a regulatory system came from? Is this mutational event able to explain where a transport protein came from? Is this mutational event able to explain where a certain enzyme came from? That's what I'm looking for. That's what I'm requiring. And that's what, when we look at every single mutation that I've ever seen anyway, we see the opposite. You've knocked the system out. You haven't gotten a new system. And so, you know, they claim some enzymes have, some proteins have formed de novo. And that becomes a whole new question, de novo. Well, what constitutes de novo? You needed one mutation. So in other words, you have a revertant mutation. It was a functioning gene. You knocked it out with a single mutation. You can restore it back with a single mutation. That doesn't count. That doesn't count. What I'm looking for is a mechanism that you would take that random piece of DNA, and now all of a sudden you start building on that, and suddenly you have an enzyme. Wow, that's amazing. How did that happen? That's what I'm looking for. And I'm not saying you have to make the whole enzyme, because obviously that's going to be something that would take a long time by evolutionary methods. I'm saying give me something that demonstrates how you've made that. Something that if I see continuing, it's going to make that enzyme. And I haven't found that yet, because in fact, it's okay, if this continues, the enzyme's going to be totally gone. There's going to be nothing left of it.
Yeah, so it seems like, I think from our observations, it would kind of go against like what you keep saying, going off the mountain, down the hill.
Yes, yes.
Climbing up.
Yes.
So question, I don't know if you can interpret this. You might know what this person is getting at here from Facebook. The second question, was there any follow-up on the case of cecal valves forming in lizards? And I guess similarly, multicellularity in microbes. So a number of questions here, specialization in yeast.
Okay, I'm not 100% certain exactly where they're trying to go with that question. I mean, you say multicellularity in microbes, but are we talking about yeast, which are unicellular? Are talking about fungi? You're talking about what microbes are multicellular? I mean, yes, there's multicellularity in some microbes, but I'm not sure. That doesn't make it a multicellular system. Bacteria even exist in colonies. They like to form biofilms, but they're still not multicellular. So I guess I'm not quite sure.
I don't know if he's
where we're going with that question.
Was there ever a case of a unicellular going into multi? I don't know. Yeah, exactly where it's coming from. But
I mean, you have some that are programmed. They go back and forth, but that's already, you know, but where'd that program come from? See, again, that's back to give me a mechanism through evolution that made that program. It's already there. All I'm seeing is mutations that knock it out. So. Yeah, but the idea of the transition from single cellular to multicellular, that you'll find a lot of papers written on that, and they're all just, you know, well, we're not sure, maybe this, maybe that, maybe this, because they don't know, because going from single to multicellular, that's a whole other level of biological complexity.
So it sounds like, yeah, we could go on and on. Lots of great questions. Love it. Maybe this first one, since I think from Christian perspective, we look at the disease suffering around us, even like SARS-CoV-2, what it does, COVID. The first question, can you discuss how parasites and viruses might have originally been created by God for good purposes, and then through mutations and adaptation took on more malevolent form?
There are several creationists who this is their specialty as they look at this. I'm not one of them, so I cannot give you nearly as good of an answer as they can give you. Just want to point that out. Seems like sometimes the idea is if the speaker can't answer a question, that means it's unanswerable. That's not true at all. But yes, there's very good reason to think that viruses and other parasitic systems actually had benefit. Some of them even still do. Frank Sherwin at Institute of Creation Research presents a talk where he goes through some of the benefits of having certain parasites. It doesn't sound pleasant to me, but actually in certain environments, you're actually benefited if you have this parasite. So we can see, although, you know, as Apostle Paul talked about, in a mirror dimly, but we can see back in the past and say, wow, it's very possible that these had certain beneficial functions for humanity prior to the fall and prior to where there became the curse, you know, and the creation wasn't functioning the way it was supposed to anymore. Viruses, in fact, let's go to modern time. When they're delivering that vaccine into your muscle, it is a virus. That vaccine is contained in a virus, which will then attach to your cell and infect your cell and put that RNA or DNA, depending on if it's an RNA or a DNA vaccine, into your muscle cell. So we are taking viruses in their basic ability and we're engineering them. So the virus in that respect then could be considered to serve a useful function because of how we're using it. So there's no reason to say that in its original form, God wasn't able to do the same thing. He made viruses to be able to deliver certain genetic capabilities to people in a manner that benefited them at that time. And since, of course, the curse, since the fall, then degeneration has occurred. And some viruses are fairly nasty now. You have to recognize the world's covered in viruses. Where you're sitting, there's some 700 billion viruses. So you're covered in viruses, but they leave you alone. They leave you alone. There's just a few viruses that actually are nasty. Just like most bacteria, you're covered in bacteria. Most bacteria, they actually benefit you. But a few of them, something's gone wrong, and now your body reacts negatively when they get inside of you, and we call them pathogens. So again, most viruses and most bacteria still have some of their residual role from creation where they either benefit you or they're benign to you. You're not even noticing they're there.
Right on. So yeah, I don't know exactly where to stop, but I think maybe one of the, I know there's the arguments for evolution is because they know that just random mutations not going to do anything. So they start coming up with, this related to the second last question here, that duplicating genes, innovating them to create new genetic information, I guess they're lying dormant. They invoke orthologs. I don't know, maybe you can explain that a bit more. Yeah, discuss a bit about that and how that's not really going to provide a solution.
Well, that comes back to, again, what does a mutation do? All right, certainly one of the models or mechanisms that evolution proposed is you had a gene that inadvertently got duplicated. Well, you only need one copy. So the other copy is free to mutate and not harm the organism, presumably. Obviously, the mutations could, but, you know, as a population, not harm the organism. And so when it's free to mutate, it may mutate and suddenly you have another gene. You have a different gene. Well, again, it comes back to, are mutations actually able to do that? Does a mutation convert gene A into gene B? And if it does, what actually transpired? What allowed that to happen? We may find that it converted to gene B simply because it knocked out part of gene A. And in fact, we find that that's actually already there to begin with. In other words, the cell already had another gene B. We may find that, oh, if we knock out part of gene B, this happens in the cell. It's not necessarily negative, it's not necessarily positive, it's just something that happens. But it comes back to, what is the mutation doing? Whether you've got a duplicated gene or you have a whole line of junk DNA lined up to be mutated, what's the mutation doing? Is it giving you the type of genetic changes necessary to account for climbing the mountain? And that's really what it keeps coming back to. Are mutations giving you this? And let me add this caveat in. Let's go ahead and say that somebody does find a mutation that even I say, wow, you are right. That mutation built that gene. That's amazing. Okay, so now you have one mutation in the tens and tens and tens of thousands of mutations that we've already documented that don't. And your one mutation gets lost in the whole, so you've gone down tens of thousands of steps to go back up one. How long does it take you to climb the mountain at that rate? And the answer is, of course, you never will. So even if it is found that there's a mutation that Dr. Anderson even has to concede, that does provide a mechanism. It's one of all the tens of thousands that don't, and it'll be lost in the gamish.
Yeah, I think there's a thing called Haldane's dilemma, like the fixing of them.
Yes, right. That how long does it take you to fix the mutation in the population? And of course, Haldane was also trying to understand, you know, you got the good, the so-called benefit mutations, the negative mutations and such. But yes, he was trying to calculate how long it would take to fix the mutations of the population. And generally what you find is that even the mutation that has a benefit gets washed out most of the time. It only gets fixed every once in a while. And that just adds to the waiting time problem. So it actually makes the waiting time problem even worse, because you may get a beneficial mutation that helps you become a human and it's lost.
Like for Dawkins' argument about the selfish gene, it's kind of irrelevant. Like it's
well, the selfish gene is very laden in junk DNA mentality. In other words, you're loaded with all this DNA that doesn't serve you any function. It's just DNA that's gotten in there over your evolutionary past. But as we see, the junk DNA argument is falling by the wayside, because more and more of what was considered non-useful, non-functional DNA actually does have functions. It actually does contribute. And so it's not selfish. It's part of your design genome.
So I think we're going to wrap it up. Maybe one more question, and I'm going to close with some more information. Maybe that first one again. I don't know if you can make sense of it. If the instructions for chromosome kissing are in the DNA code currently, couldn't an addition or substitution to pre-existing code create the code for that?
Well, again, I don't know how. I mean, yes, in theory, on paper, I suppose, but in reality, if we look at what insertion and deletion mutations actually do, they don't do that. So I have yet to see them be able to provide a mechanism that would account for the kind of chromosomal interactions that we see in our nucleus. So yes, on paper, anything is possible. On paper, I can do all kinds of doodling and come up with all kinds of ideas and make all kinds of genetic changes. That's one of the things when I used to work in a molecular genetics lab. Well, on paper, we could design all kinds of vectors. Now, getting that vector to actually exist, in other words, actually trying to make that vector, that was a whole nother challenge. And so, yeah, on paper, we could say, well, if we had a deletion here, a mutation there, maybe it'll work, but, you know, does it go back to what happened in real life? We find, well, that didn't work after all. And so, again, I don't know of any addition or mutation, mutation or deletion mutations that would enable that kind of chromosomal interaction in a cell.
Right on. So I think there's some questions that may not been directly related to the topic. So maybe I could come back tomorrow for Dr. Kevin Anderson's talk about dinosaur soft tissue.
It'll be a
dating.
Totally different topic.
Yeah, yeah. So yeah, there's a question here about dating. We won't get into it here. And then maybe hopefully some that would have been answered. If not, we'll try to keep your answers and your emails if you provide that.
Yeah, and of course with Daniel's question, yes, as I mentioned, by and large, scientists will acknowledge that natural selection doesn't create anything. But then when you listen to them talk about evolution, you kind of think, well, okay, I thought you just said it because they tend to then talk in terms of natural selection creating things, in part because they don't know how else to explain it. And so there seems to be at odds with what they know to be the case of natural selection versus what their power they're trying to give natural selection to do. And so it's always kind of an interesting conundrum of wording that you'll find themselves in when they're talking about natural selection and evolution.
Yeah, and I want to get back to also like you're describing the question of like whether we came from a population of many humans or two, like the evolutionists don't even want to consider if you start from a biblical view or like if the two Adam and Eve had different genomes to begin with, like there's always going to be, there can be a different interpretation if you start with different set of starting assumptions. Like if you mutated quickly and you fix the population, like there's all other aspects of it too. So
well, and some things to consider in that, because that is a popular idea that, well, we can't start with two. We got to start with a bigger population than that, and then to go through a lot of bottlenecks. Mitochondrial Eve is very consistent with starting with two, because she's the mitochondrial mother of all contemporary humans. Now, the argument will be, well, but there were other women. We don't have any mitochondrial evidence that there are other women. That's just a speculation because you need that for evolutionary argument. But genetically, there's no evidence mitochondrially of another woman. There's just the one, all right? You can't make quite as clean of argument of the Y chromosome because of the nature of the Y chromosome, but there is pretty good evidence to say there was a Y chromosome atom. So again, say, well, but there was other atoms, but there's no genetic evidence of another atom, at least from the Y chromosome. And then if we go to the rest of the genome, all the other chromosomes, the 1 through 22, our genealogical, now this is different than our genetic, recognize that, but our genealogical ancestor lived about 3000 BC. And that's published, that's accepted. What that's telling you is that what we have is such a small population. Again, that's totally consistent with an original pair. So when they're talking about, well, we can't have original pair, that's just imposing an interpretation, because the data in no way requires that we can't have a original pair. It in no way requires that. And so let's start with original pair and see where we get. Voila, we're just fine genetically. It doesn't create any problems, at least not what they're trying to point out it creates.
Right. So yeah, thank you, Dr. Kim. I think we'll close it there for tonight and have you back for tomorrow. I just want to go over, share my screen and go over some of the things to get more information. So thanks for joining us, for all of you who did and even stayed this late. Just share this. I want to bring your attention to our website, creationbc.org. Find more information about us. Also, you can link to the Facebook group if you haven't already joined us through there. There's a link there. So here you can find more information about, say, upcoming speakers. So at the end of May, we hope to have another Ph.D. Scientist give us talks, Dr. Jerry Bergman. We can hopefully finalize those dates soon. So look to our guest speakers link up here. There's also, if you want to be put on a mailing list to be informed of when we do have our next talk, just fill out this form and we'll add you. Promise not to spam you or sell your information or anything like that. I want to, again, bring to your attention, there's the Echoes of the Jurassic, the significance of dinosaur fossil tissue. So join us tomorrow, 1 p.m. Pacific time, Pacific Daylight Time with Dr. Kevin Anderson again. And then also just want to bring to your attention, this is just kind of tip of the iceberg. There's always more. If you're a student, if you have an interest, love for science, you're talking to other people, they always have objections, get informed. We have many resources for you to do that. If you liked the talk tonight, maybe consider getting Beyond is Genesis History, Volume 2, because in there, it features 2 talks by 2 sessions with Dr. Kevin Anderson, one of them about what he talked about today, maybe a bit some of that. So you can pass that on. Many good creation scientists, again, all PhD level in their respective areas of expertise, commenting on the biology and life, essentially. If you have children, consider something like the Answers book for kids. There's 8 volumes. It's for typical questions kids would ask about creation and science. Each individual one is only $8. There's also, if you go around to museums, you don't have people or the museum displays showing that evolution is really more like stasis. Their animals really haven't changed over supposedly hundreds of millions of years. And many animals that would have come to a shock for people to find out they lived with dinosaurs, for instance. So this Dr. Carl Werner went around visiting many museums and documented it all. I strongly recommend getting this grand experiment. There's episode 1 and episode 2, this DVD and in book format. And then, just to get if you're new to all this and you need an introduction, maybe older kids too, the great book is Creation Basics by Institute for Creation Research with many colorful photos. But just, come visit us on our store. Check us out if you want to become a member, and you can have access to our free DVD lending library as well. So just to be equipped, be prepared to share the faith, the hope for the reason that you have. Tear down those arguments that the unbelievers may have. And we hope that we can get in touch with you and keep informed and keep in contact and keep this dialogue going.